Assalamu alaikum. In today's lesson, we are going to discuss in detail about the processes of transpiration and translocation. But before we move on to that, let's talk about the minute details about the structures that are involved in the type of water transport mechanisms that will be required. So first up, let's talk about the root hair cells. So here you are looking at the root of a young radish, and then you can see actually the root hair like emerging from both the sides, and you can see there are a lot of those, and these are visible to naked eye as well. So these root hair cells are actually specialized, and they are exchange surfaces for the uptake of water mineral ions. So you can see like in the soil, definitely we have water and mineral ions, and they are actually, these root hair cells are going to increase the surface area for absorption of these. So these root hair cells are about 200 to 250 micrometer, and this makes them visible to the naked eye. And with thousands on each root, they actually maximize the surface area. So you can see there are a lot of there's only one root that they've shown you, right? And you can see there are a lot of root hair cells emerging on both sides. All right then. So here let's talk about the mechanism of water absorption. So we know, okay, in the soil we have water and the mineral ions as, there, and they're actually found in the small air spaces in the soil, right? When these roots, as they make contact with the soil, so first what happens, the minerals, they actually get inside the root via active transport, right? So because of that, what happens, the concentration, the solution here in the cytoplasm and vacuole actually becomes more concentrated. And this actually reduces the water potential here, right? So because of that, what happens, since water is like water in the soil is actually at a high water potential, it would diffuse in, right? Because of this concentration gradient. So because of there is more concentrated solution in the cytoplasm and vacuole now, water will flow in from the soil via osmosis now, right? So this is how water is going to enter the root hair cell. Now here we are looking at actually the pathway of water movement from root hair to xylem, right? So if you focus on this diagram here, so here they have shown your root cell, and you see okay, water has entered through osmosis, and now the blue arrows you're looking at actually is showing you the direction of water movement, right? So this is how water is moving. So it enters the root hair, then through the root hair, it would enter the epidermis, which is the first layer of the root. So you can see here as well. Okay, the first layer, epidermis, then it crosses the cortex, which has a number of cells here. So you can see this part here that they've shown is the cortex, right? Then comes the endodermis, right? And so here you can see this layer, endodermis, then comes the pericycle, right? Which they have shown you here. After the pericycle, we have the phloem, and then we have the xylem. So you can see here, these are the phloem and the xylem. So this is the transfer section of the same, right? And here they've shown you how the water moves from the root here, and eventually it crosses the epidermis and the cortex, endodermis, pericycle, and then finally it goes to the xylem. So this is the direction of water movement. All right then, so let's talk about these different pathways that actually water will take to reach the xylem. So the thing is, water moves through the root cells into the xylem tube by three pathways, right? In the syllabus, they have mainly focused on two, apoplast, and symplast. Vacuolar is more or less the same as symplast, right? It's just the difference between that the water is actually passing through the vacuole instead of passing through the whole of the cytoplasm. Since you know vacuole is there in the cytoplasm, it's there in the cell, inside the cell, right? So this would be passing through the vacuole only, right? So these allows the plant to get water fast. Like basically, the main purpose is to get water as fast as possible, right? So these different pathways that actually will be adopted or adapted by the roots. Let's see what happens. So first up, we're looking at the Apoplast pathway. So Apoplast pathway is actually as the water is moving along the cell wall, right? So again, one way to remember is that Apo starts with A. So you can just remember it moves along the cell wall. So actually, but biology, when you're looking and using the term, so you say it goes through the cell wall, right? So if you focus on these numbers, 1, 2, 3, and 4, so like first of what is happening, water enters the cell wall. So you can look at the blue arrows, right? Then it moves through the cell wall. So you can see the movement. Let me use a laser pointer here. So you can see water enters, then it moves through the cell wall. So this is cell wall. Then it can move from cell wall to cell wall, definitely across the intercellular spaces. So here you can see it is moving across the cell wall, right? And then you can see one pathway here as well, right? And then between the cells, we have these intercellular spaces, right? So it passes through those and then it can go to the next cell, right? And if the cells are actually, let's say, connected at these points, these are like together, so it will pass from one cell wall to the next. And if you see spaces between the two cells, so definitely there will be an intercellular space there. So it will go from one cell to the next cell through the intercellular space, right? So this is the Apoplast pathway. And we just discussed, yes, it is a movement of water through the cell wall and the intercellular spaces, intracellular spaces rather, right? And then what is happening here is that water, it is moving because of cohesive and tension forces. Cohesive forces are actually, if you recall from a discussion in biological molecules, these water molecules are actually joined by hydrogen bonds, right? So they stick together. So these sticking force, like they are actually joined together to each other, as well as they would adhere to the walls, the cell walls as well, right? As they adhere to the cell wall and they're connected to each other as well, this would create a tension, right? So as the water leaves from here, right, this would actually pull the other water molecules along with it, right? So it actually makes a water column. So these cohesive and tension forces, they act on the cell wall and then they pull the water up the plant, right? So these are two forces involved, cohesive and tension forces, right? So it is basically water particles are joined to each other through the hydrogen bonds, as well as the water particles connected to these walls of the cell wall as well, right? And then this actually pulls the water sort of up the plant. So this is the Apoplast pathway, right? And for this, it is said this is the fastest movement of water, right? Fastest as such because there's no hindrance as well. It's just the cell wall and it is passing through it, right? Since if you just compare it with what would happen in the cytoplasm, there are many cellular organelles as well, right? So definitely it would take for water to take some time to go from one cell to the next, right? But cell wall wala jo pathway hai, Apoplast pathway hai, it is the fastest movement of water because there are no hindrances there. Okay, then coming to the Symplast pathway. Symplast, just remember that here, the water is moving through the cytoplasm. Okay, so Symplast mein, it is moving through the cytoplasm, and right now it is moving through what we say is it is moving through the living spaces of the cell, right? So here you can see again from point 1 till 4. So if you look at point number 1, so you're looking at water enters the cytoplasm, but it has to cross the cell surface membrane as well, right? So it enters the cytoplasm across the partially permeable membrane. Then it can move up the sap. So it depends. It can move across the vacuole, right? Or it can take the other cytoplasm pathway as well, right? So again, there's the reason I said that vacuolar pathway is like almost the same as Symplast. Usme basically we are focusing on only vacuole ke through water move. Okay, so water can move up into this can move up into the sap in the vacuole to the tonoplast, right? Because it will have to cross the tonoplast as well. Water may move from cell to cell through the plasmodesmata. Then you can see that cells are actually joined to the other through the plasmodesmata, so it can cross the plasmodesmata and reach the cytoplasm of the other cell, right? And water may move from cell to cell through adjacent plasma membranes and the cell wall. So we just see this as well, right? So it can go through that. So it can adapt all these 3 different pathways, right? So each cell further as it goes away from the roots, okay? As it is going away from the roots, so what is happening? The water potential will actually will keep on becoming lower, okay, in these cells as they move away from the roots, right? And since like these cells here are like, let's say, close to the roots, so water potential is higher here. So what will happen? So because of this pressure or potential difference as well, water will move from high water potential area to the area where there's a low water potential. Okay, so this is also what encourages the movement of water along the Symplast pathway, right? And the vacuolar pathway, as I said, it's the same as the simplest pathway. It is simply that water is moving through the cell's vacuoles, right, in addition to the cytoplasm, right? But for this, it's like it is the slowest pathway because again, the vacuole content the cells have, and you have a lot of like solutes there as well. Okay, so definitely it will create a pressure. So this is the slowest route of water transport. So here you can look at another diagrammatic view of all the three pathways of water. So the red that you're looking at, so the red one that we're looking at is the Apoplast pathway. As you can see, it is the water is moving through the cell walls. Okay. So if you follow the red arrow here, if you look at B, this is the Simplast pathway. So you can see what is moving through the cytoplasm, then it crosses the plasmodesmata and then goes to the next cell. Then again crosses the plasmodesmata, crosses another cell. And here they have shown you the vacuolar pathway, right? All right, and this is another view that they've shown you. So the blue arrow that we're looking at is the Apoplast pathway, right? So they've shown you these pathways across the route right now. Okay, and here you're looking at the Simplast pathway, right? All right, and the next thing that we have to discuss is about the Casparian strip, right? So this Casparian strip is actually found in the endodermis, right? So if you remember, the first layer was the epidermis, right? Then is the cortex. Right now we're talking about the root, right? And then becomes the endodermis, and this is the Casparian strip. And this Casparian strip is made up of a material which is impermeable, and it is known as suberin. Okay, this is a waxy material and it is impermeable to water, right? So as water, you can see, okay, these are the soil particles, this is the root hair cell, and as water moves in, right, through osmosis, right? So it crosses the epidermis and the cortex, but when it comes here at the endodermis, it cannot follow the apoplast pathway, right? Because in apoplast pathway, the water passes or moves from cell wall to cell wall. This suberin layer is actually surrounding the entire cell, right? So water cannot use the apoplast pathway in this case. In this case, water actually is forced into the simplast pathway, right? And eventually then it will go into the xylem, right? So this is one barrier there, right? So it has to follow the simplast pathway rather than the apoplast pathway, right? So this is known as a Casparian strip, and it is impermeable to water, right? And the material is known as suberin. This is another view. So here you're looking at the soil particles, is the root hair cells, right? And the water enters the root hair cells, okay? And then you can look at the movement, right? Another view of the Casparian strip. So here you can see, since it is actually like encircling the entire cell, so water cannot follow the apoplast pathway at all, right? It must pass through the simplast pathway. It must pass through the cytoplasm. This is another very good diagram that actually would help you in understanding. So the blue dot here shows you the apoplast pathway through the cell wall. So you can see if you follow the arrows here, right? So this is the epidermis. So okay, if you follow the apoplast pathway first, so it follows the cell wall, then it goes to the next cell, right? Which are here in the cortex. Then it goes into the endodermis. But endodermis, maybe have this Casparian strip. So water cannot cross the cell wall in this case. Now it will have to go inside the cytoplasm. So it has to cross the plasma membrane. It has to go into the cytoplasm. And then from the endodermis, it can follow the same route through the plasmodesmata, through the pericycle, and then later on to the xylem, right? And if you look at the simplast pathway, so you can see, right, it crosses the cell wall, the cell membrane, then it goes into the cytoplasm, and then it has to go through the plasmodesmata to the cortex. Then it again crosses the plasmodesmata, goes to the endodermis, right? So there is no barrier for the simplast pathway, right? And then the pericycle, and then to the xylem. And then again, okay, also you can look at this is the root hair cell that they've shown you. So they have shown you apoplast pathway following cell wall to cell wall here, the one in green, right? So you just saw, okay, it crossed the epidermis, then whole of the cortex, then comes the endodermis, but endodermis with the red strip that you're looking at is the Casparian strip. Okay, so it will follow the simplest pathway after that. and then it can go into the pericycle and then definitely into the xylem, right? And here they have shown in the Simplast pathway. So you can see from cytoplasm to cytoplasm, right? And here there is no barrier. यहाँ पे Casparian strip is not blocking the way, right? So Simplast pathway will keep on actually continuing, okay? But Apoplast के अंदर there will be a barrier. So Casparian strip will not allow water to follow the Apoplast pathway. So it will be forced to follow the Simplast pathway. Okay, it happens at the endodermis. All right then. For this, I want you to have a look at an animated video about all these pathways, and then we'll move on to the topic of transpiration.
Trees must transport water and minerals to their highest leaves. How does this happen? Interestingly, trees move water by losing water. Evaporation of water from plants is called transpiration. Water diffuses from inside a leaf to the outside air through pores. Inside a leaf are air spaces. Water evaporates constantly from the surfaces of surrounding cells. To better see the water, let's show it in blue. Notice that water forms a film on cell walls, including the walls of cells in leaf veins. Within the veins are hollow, tube-like cells, shown in brown. These cells, also found in stems and roots, are part of a tissue called xylem. Water and minerals are transported from roots to veins through the xylem, and then from veins into the walls of other leaf cells. What maintains this flow? Let's look at the surface of a plant cell. A thin layer of water covers the cell wall. As water evaporates, the cell wall begins to dry out. Look at how water sticks to the cell wall. This is called adhesion. Water molecules also stick to each other. This is called cohesion. Cohesion and adhesion are the results of hydrogen bonds, the dashed lines. Because of cohesion and adhesion, transpiration creates a tension from leaf cells through the xylem to the roots that pulls water molecules up the plant. Now let's look at how water and minerals enter through the roots. Roots are highly branched, creating a lot of area for absorption. Root hairs near each root tip increase the surface area. Water and minerals, shown in yellow, can follow two paths from cell to cell within a root. In one path, they travel in the cytoplasm, which is connected between cells. In the other path, they travel within cell walls. In the path within cell walls, however, they encounter a waxy barrier that blocks entry to the xylem. To continue, the water and minerals must cross a plasma membrane. In this way, only certain minerals can enter the xylem. After moving into the xylem, water and minerals are pulled up to the leaves. Water evaporates from leaf cells into the air spaces inside leaves and exits through pores. And that's how water can reach the highest leaves of a tree.
So transpiration is the loss of water vapor. So let me write it here. It's not loss of water. It's the loss of water vapor from the leaves of a plant. And most of this occurs from the underside of the leaf, right? But there are many stomata in the epidermis, right? So here you can look at the guard cells, right, which are surrounding the stoma. So most plant cells control their water intake by opening and closing the stomata, right? So this happens as the water level changes in the guard cells, because guard cells are actually surrounding each stoma. So the water level in the guard cell actually changes. This allows the stoma to open or close, right? So this water level change is actually being done passively by osmosis or by the active transporter solutes, right? And besides this reason, opening or closing of stoma, transpiration rates are also actually dependent on other environmental factors as well, right? So these could be any change in temperature or humidity, right? So these changes actually will also affect the rate of transpiration. So first, let's see what actually these guard cells are doing, or what's their role in this transpiration, right? So when we talk about the opening and closing of stomata, how this opening and closing is being regulated, the stomal movement is actually the result of changes in the tertiary of the guard cells. So remember, it is the water level or change in the water level that is taking place in the guard cells. This is what is causing the stomal movement, right? If the water flows into the guard cells by osmosis, right, their tertiary would increase and they would expand, right? Now, what will happen because of that? These guard cells actually have inner layers, right? And they have this outer layer, right? So the inner layers are actually quite thick, right? So they would actually would not budge, right? Inka size as a change nahi hoga, but the outer wall are thinner. So these would actually elongate, right? So this is what actually causes the stoma to open or close, right? So as the inner walls are relatively inelastic, it makes these guard cells bend and then draw away from each other, and this opens the pore, right? So this is what happens. The pore is opening because the water is entering the guard cells, increasing the turgidity, and this is what is like causing the stoma to open. Okay? Inversely, when the water will leave the guard cells, right, this will cause the stoma to close, right? So when the stoma is closed, we use the term flaccid, right? So when the stoma is closed, it is because the water has moved out of the vacuoles, right? Because of osmosis. So here you can see actually a closed stoma right now. Okay, the opening here is closed, right? So as water moves by osmosis, okay, so what is happening? We know the outer wall is thinner, okay, so it will be easy for it to actually bend back, and this will close the stoma. And when the like the guard cells have taken in water, or the water has entered by osmosis, this will increase the turgidity, right? And what will happen because of that? Since again, we have already discussed the outer wall is more flexible, it is thin. Okay? So this will cause the cell to bend and this will open the stoma, right? Inner walls are elastic. Okay? They do not give in. Okay? They do not bend. It's the outer wall, okay, which is actually causing the stoma to like close or open because it is thinner. Okay? So this is when we call case when the cell is turgid, okay, it will cause the stoma to open. Okay? And when it is flaccid, it will cause the stoma to close. Right? So here you can look at another diagram, okay, of the guard cells and the stoma. Right? So here you're looking at case turgid, right? Water is entering the vacuoles. Actually, they take in water, the osmosis, and then you can see it like increases the turgidity. The outer wall will actually bend, and this will cause the stoma to open. Okay? In this case, water is actually moving out by osmosis, right? And what you can see because of that, again, is the outer wall that bends back and the stoma closes, right? All right then, so that's much about the stomata. And so how does water move up the stem? So when we talk about the water moving up the stem, there are three processes that we need to take into account. So first of them actually is cohesion, okay? Then adhesion, and then comes the root pressure. Achha, when we talk about cohesion, we are not talking only about cohesion, but it's actually together cohesion, okay? Cohesion tension together, and the term that will actually be used for that is transpiration pull. Okay, so these events will happen, which will actually cause the water move up the stem. So transpiration pull is cohesion and tension. Okay, both of these together, cohesion tension. Okay, then comes the capillary action. Capillary action is adhesion. Okay, we are going to talk about all of these in detail, right? And then comes the root pressure. Root pressure is an active process, right? So these three events have to happen in order for water to move up the stem, right? So let's talk about the transpiration pull or this cohesion tension theory. But for that, we need to understand first how these hydrogen bonds are actually made among these water molecules, okay? Because this is what will actually help you understand this entire process of transpiration pull, right? So we know that many of the properties of water are actually due to its ability to form hydrogen bonds. And how are these hydrogen bonds are made? So you can see there is oxygen, there's a slightly negative charge on the oxygen, and there's slightly positive charge on the hydrogen. So these are individual water molecules. And then you can see, since there's a like slightly negative charge here on the oxygen and slightly positive charge here on the hydrogen, there will be actually a bond made between these two, which is known as a hydrogen bond, right? So here they have actually shown you two water molecules. But if you look at this diagram here, right, because water molecules, like there are a lot of water molecules, so water molecules, they stick to each other, not just like to the molecule next to it, but the molecule which is like below it or above it as well. So this is actually what like constitutes this cohesion between the water molecules. So since water is a polar molecule, so we have already discussed it has a slightly negative oxygen atom and a slightly positive hydrogen atom, right? And these charges are not evenly distributed, right? So what will happen in the xylem, water molecules spontaneously will arrange themselves, right? They will arrange themselves spontaneously. So what will happen? The positive and negative charge poles lie next to each other. So here you can look as well through these dotted lines, you can see these are hydrogen bonds made here, then made here, then made here. So they're sticking to each other, right? So this causes all these molecules to cohere to each other or stick to each other. So what will happen, as some of them are leaving the plant by transpiration, and we talked about that, the transpiration is the movement of water vapors through the leaves of the plant, okay, basically through the stomata. So as the water molecules are leaving the plant, others are being pulled up behind them, because again, see, they are connected to each other, they are joined by hydrogen bonds. ठीक है, since they're connected, अगर ऊपर से move करेंगे, so these all will actually move as a water column, वो उसको pull करेंगे. That is what we call as a transpiration pull. Okay, so this is what we call, this is what is actually creating this tension here. Okay, so it's cohesion and tension both. Okay, so this was cohesion tension, right, because of the hydrogen bonds. Now coming to capillarity, as we talk about the addition of water, so water adheres to the side, to these xylem walls actually, right? So this addition of water is actually what we call as capillarity. Okay, so addition of water is when water sticks to the other molecules, right? So cohesion kya tha? When water is sticking to the other water molecules by hydrogen bonds. वो चीज थी cohesion. Addition kya hai? When the water molecules are sticking to the other molecules. Like in this case, you're talking about water adhering to the xylem walls. Okay, the xylem walls, they have lignin, they have cellulose. Water वहाँ जाके stick हो रहा है. So here you can look at this diagram as well. So here they have only shown you addition. Right, so you can see water molecule is sticking to the walls of the xylem. ठीक है, this is addition. और cohesion क्या होगी? जब ये आपस में भी एक दूसरे के साथ connect हो जाएंगे. So if I could just draw a line, so see, if this is for example oxygen, ठीक है, so it will attach, let's say, to water here. ठीक है, so this will यहाँ पे इस तरह करके hydrogen bonds बनना शुरू हो जाएंगे. Okay, so this is addition. Water molecules connecting to the walls of the xylem, and cohesion होगा when they are connected or sticking together, making hydrogen bonds. Now, the thing with xylem is that the narrower the xylem, the bigger will be the impact of capillarity. Up here in this diagram, it is just to show you the difference. Okay, if you look at this test tube here, which is actually inserted in water beaker. Okay, so you can see actually water, even though the lumen is wider, ये moderate है, and this one is a thinner here. So okay, water has this capacity, it has this tendency to adhere to other molecules. Okay, to other molecules. In this case, it is glass tube. ठीक है, तो water molecules stick हुई इसके साथ साथ, but then you can see and they are connected here, cohesion से एक दूसरे से connected है and to the wall. But now still, even though this test tube has a wider lumen, फिर भी a little of it actually has moved up. ठीक है, क्योंकि water यहाँ पे stick करता जाएगा and it will move up. ठीक है, but then जितना ज़्यादा lumen narrow होता चला जाता है, you can look at the distance here. ठीक है, and then look at here, because now here what is happening? ठीक है, it has more surface to get attached to. ठीक है, यहाँ पे the lumen is narrow, so water molecules को ज़्यादा surface attach होने के लिए, so they will actually move up. So this is what is known as capillary action, something that you see in the straw as well जब आप juice पी रहे होते हैं. So actually it like quickly goes up. वो capillary action की वजह से it goes up. ठीक है, so this is known as capillarity or adhesion. ठीक है. The third thing that we have to talk about is the root pressure. So remember we talked about the three processes which are actually working together. So one was the cohesion tension theory. Okay, that was the transpiration pull. Second, we talked about the capillarity, which is the addition. Or third that we have to talk about is the root pressure. Okay, so root pressure, actually think about it as, take it back to when we were discussing about how these solutes are actually entering the roots of the plant. Okay, so since like in the soil, there's water as well as the minerals there. So these solutes are actually actively transported into the roots of the cells. Okay? They are being actively transported. So what is happening because of that? It actually reduces the water potential inside the roots. Okay? The water potential वहाँ कम हो गया. So what is happening? It causes the water in the soil, which is considerably at a higher water potential. It will enter the cell through osmosis. What is happening because of that? Now this is increasing the hydrostatic pressure in the root. So root के अंदर जो pressure है, it becomes positive there. ठीक है? So this creates a positive pressure. ठीक है? And this pressure, because roots में pressure ज़्यादा हो गया, ठीक है? और ऊपर की तरफ if you up the plant, ठीक है, तो ऊपर pressure कम होगा water का as compared to down below. So this will actually force the water to go from the root. ठीक है? So pressure जो है water will go from the root upwards. ठीक है? ऊपर की direction में flow करेगा because there's a positive pressure down below in the root and there's a negative pressure up in the stem. So naturally water, since it moves from high water potential to low water potential, to water ऊपर ही move करेगा. ठीक है? This will force the water up the stem. ठीक है? So this is what we call as the root pressure. So all these things together, ठीक है? These are together in actually bringing about transpiration. ठीक है? So when we talk about the movement of water up the xylem, so remember these are the three things: cohesion, which was water molecules sticking together by hydrogen bonds. Okay? And then as they're sticking to each other, remember, so water molecules are sticking to each other side by side, as well as water molecules below them and water molecules above them. So transpiration के through जब water remove हो रहा है, so it is actually pulling the water molecules below it. It will actually move as a column. ठीक है? Then addition, this water molecules connecting to the side of the xylem walls, ठीक है? Side of the xylem walls. So it will be attached to the xylem as well. So, ठीक है? So that is addition. And then comes the root pressure. ठीक है? And says as water comes in by osmosis through the roots, वहाँ पे root pressure बढ़ जाता है. Hydrostatic pressure is increased. And all of these working together actually brings about the movement of water up the xylem. And all of this is actually what we call as a cohesion tension theory. Right? So again, if you look at this diagram here, okay, so here they have shown you what is happening at the root level, what is happening here at the stem, and what is happening here at the leaf, right? So you can actually go through these points. Okay, so when we talk about the cohesion and addition, okay, so cohesion plus addition, both of these, because cohesion is between the water molecules. Okay, addition is when these water molecules are sticking to the walls of the xylem. Okay, so both of these, okay, they keep the water in the xylem vessel moving as a continuous column. Okay, and you can see these vessels are full of water. You can look at the arrows, like you can look at this direction. So they're full with water. Okay, and what is happening here, because xylem, if you recall the structure of the xylem vessels, okay, these are dead cells. Okay, there are no end plates, and there's no protoplasm to actually obstruct the way of the water. Okay, so water will keep on going up. Okay, and since water root cells, it was actually entering through osmosis, but this is not the case here in the xylem vessels in the stem. यहाँ पे ऐसा कुछ नहीं है. ठीक है? यहाँ पे no osmosis is occurring here because यहाँ पे तो कोई solutes है नहीं. There is nothing here that is blocking the way. ठीक है? So there is actually so it will keep on moving up. ठीक है? The column of the water in the xylem vessels is there, so therefore under tension. ठीक है? So there is stretching force. It will keep on going up. ठीक है? As transpiration goes through, it is leaving the leaf. ठीक है? You know, because there is a transpiration pull, so it will keep on going up. Right? Now here actually let's talk about the whole process, like what is happening at the leaf. Okay, transpiration stream, okay, how it is actually leaving the leaf and what is happening to the movement of water along the xylem. So if you focus here, so here they've shown you the stoma. So as water is evaporated, or in fact water is evaporated in the form of water vapor. So what is happening in this case, what would happen like here is as water is leaving, so water pressure here actually will become low. Okay, so these are the spaces, there's spaces between the mesophyll cells. Okay, these are the air spaces. So air spaces में water pressure becomes low. Okay, or water potential becomes low rather. Okay, so what will happen? These cells which are adjacent to these air spaces. Okay, अब यहाँ पे water potential ज़्यादा है. Okay, so water will move out of these cells into the air spaces. ठीक है? अब as the water is moving out of these cells, यहाँ water potential कम हो गया. So then you can look at the xylem vessels here. Water potential is more here. So water will move from the xylem into the cells. ठीक है? So you can see how actually water will be continuously kept on being pulled. ठीक है? So even if it is being lost here as a water vapor, ठीक है? It will actually be continuously it will actually xylem ke through this transpiration pull actually will keep on happening actually. देखिए tension हमेशा रहेगी यहाँ पे, right? So water evaporated from the leaves, ठीक है, through the stomata. It decreases the water potential of the air space inside the mesophyll, ठीक है. So when the water potential reduces here, this causes the water to move into the air spaces from the adjacent cells, ठीक है. So adjacent cells से water यहाँ पे move कर जाएगा, ठीक है. So as the water here in the adjacent cell decreases, water will move out of the xylem into these cells, ठीक है, in the leaves, ठीक है. And we know like xylem can like what there's cohesive forces between the water molecules. Hydrogen bonds are already there. Then we also know there is adhesive forces. Water molecules are attached to the xylem vessels. Okay, this will actually create the tension, and this will all result in capillary action, right? So as we are talking about the water movement, so remember, वो Apoplast pathway भी हो रहा होगा. It will be moving through the cell wall, and symplastic pathway will also be in play. Okay, so this is how water is pulled up the plant, right? Or this is what we call as a transpiration stream. This is another diagrammatic view. So here they have shown you as well the route as well. Okay, so how water moves in through the apoplast or symplast pathway, okay, up the xylem, and then you can see this is what they've shown you. Okay, water leaves as a water vapor, okay, but then because of the transpiration pull, okay, water will be kept on moving upwards in a continuous flow from the xylem, okay. So definitely through the road it will be sucked in, okay, and you can see there will be a continuous flow, okay. There is pathway ko. Nothing is actually breaking this pathway. Continuous column, it will be keep on actually moving, okay, across the xylem, okay, and then it will leave the plant as a water vapor, okay, and it will go on and on. So this is the model of water transport through a plant, right? So here you can read through it, okay. You can pause the video, you can read through it, add it to your notes. So go through it, and then we'll move on to the next slide. All right, this is another diagram. So I want you to have a close look at it. Try to design your answer. The route taken by water. So see, in this case, actually, when we're talking about movement of water, so remember we are talking about movement of water, like we use the term mass flow, because it's not just water, it is actually the solutes also which are dissolved in it, right? The minerals which are actually, they got in by active transport. Okay, so it's not just water, okay? So that's the reason when we talk about kg water upar ja raha hai, there's actually a mass flow. Okay, it's not just water, it's water plus the solutes, the minerals. Okay, and this is another view of the same, right? So vertical transport of transport by the xylem sap. So let's just have a recap. Okay, during daytime, we know the stoma is open. Okay, so stoma is open during the daytime. Uska reason basically kya hai? Because daytime में photosynthesis हो रही है. So carbon dioxide has to diffuse in as well. Okay, water is also diffusing in. But at the same time, oxygen is coming out. The water bhi ja raha hai as a water vapor. Okay, so when the water content in the atmosphere is low, it builds up a water potential gradient in between the substomal spaces in the atmosphere. So water will leave through the stomata by transpiration. This reduces the water potential of the mesophyll cells, creating a water potential gradient between the xylem and the mesophills. As the water moves from xylem to the mesophills, this builds up a suction pressure or a tension within the xylem vessels, causing a negative pressure potential. Suction pressure thrives upon up to the xylem vessels of the root. Then the water moves upwards to the xylem vessels as a continuous flow. Continuity water column is kept by cohesion and addition of water molecules and is power generated by the capillary action. Due to this, a continuous gradient of water potential is maintained from the soil to the plant of the atmosphere. This results in pulling of water continuously upwards, so cohesion, addition, and tension theory. Right, so these are different versions of how you can frame your answer. Okay, you can go back, you can look at the diagram, right? Because you get a question actually where they ask you how water is transported at the xylem. Okay, so you need to take account of all these points. Again, answer logical hona chahiye, starting from कि किस तरह start हो रहा है process, what is happening in between. So एक logical answer आपको होना चाहिए from start to finish. So एक दफा read कर लें and then we can move on. All right then. As we have talked about the process of transpiration, ठीक है, and we know okay water moves as water vapors, ठीक है, it goes through the stomata. But then we also need to think about the factors which would affect transpiration. Okay? So transpiration rate actually increases or decreases depending on these factors, right? So there are 4 factors that we have to talk about: light intensity, temperature, humidity, and wind. All right then. So if you talk about light intensity, so we know that during daytime photosynthesis has to happen, so stoma will be open. Okay? So if there is more light, it will cause the stoma to open. But think about it, stoma open hai, photosynthesis is happening, but uska like sort of side effect क्या हो रहा है कि definitely water, क्योंकि stoma open है, the water will be lost as water vapor. Evaporation होगी, you will have a large surface area for evaporation. Okay, so increase light intensity actually has a positive correlation. The more the light intensity, okay, the stomata will be like that, definitely will be kept open for the like longer time. It will cause more water vapor to be evaporated. Okay, so it has a positive correlation. Okay, now think about the temperature. When we talk about temperature, think about the like these heat energy actually causes these molecules to actually move at a faster rate. So kinetic energy increase over here. Okay, if the kinetic energy is increasing, okay, so molecules will be like moving at a faster rate. This will actually cause more evaporation. Okay, so again, more the temperature, okay, the more the evaporation will happen. Okay, it will happen. So that's a positive correlation. Higher temperature, higher the transpiration rate. Okay, more the light intensity, more the transpiration rate. Okay, even if you talk about the wind, okay, if there is more wind, it will definitely blow away the humid air containing the water vapor. So if the air is being blown, what will happen? Water potential gradient actually maintain to करना है. So definitely the more the wind, so there will be a positive correlation. Okay, so more transpiration will happen because of the wind. But if you talk about the humidity has a negative correlation. Okay, because what will happen? There is already more water vapor in the air. Okay, so what will happen because of that? It will make the water potential more positive outside of the leaf. Leaf के बाहर water potential ज़्यादा हो गया. And we know कि water potential ज़्यादा हो जाता है, तो definitely फिर इसकी movement water की बाहर की तरफ से अंदर आएगी. तो इस केस में क्या होगा? Stomach close भी हो सकता है. ठीक है, तो इसमें जब stomach close हो रहा है तो definitely transpiration is not happening then. ठीक है, क्योंकि उसका rate slow down हमें करना है. Already water vapor बाहर ज़्यादा है. और transpiration में क्या हो रहा है? Water vapor actually is water is leaving in the form of vapor. Okay, so in this case, there will be a negative correlation. So these are the 4 factors that we discuss about as we talk about the rate of transpiration. Right, so these are just a diagrammatic view of all those. So यहाँ पे they have shown you okay, this is a thick layer of moist air because यहाँ पे water vapor है collected. ठीक है, so if let's say there is still air, so still air में the humid environment it will stay like this. ठीक है, but if there is moving air, wind है, तो उस case में this will be blown away. So इसकी वजह से क्या होगा? Transpiration rate will eventually increase होना शुरू हो जाएगा. Here they have shown you a graph to show the effect of light intensity. Because the light intensity के time पे you can see the transpiration rate increase हो रहा है. ठीक है, and then it reaches equilibrium. But more the light intensity, ठीक है, it increases the rate of transpiration. All right then. We have already discussed about these factors. So we have discussed as well, the higher the temperature, the higher the transpiration rate, higher the light intensity, higher the transpiration rate, higher the humidity, however, the lower the transpiration rate. Okay, and more the wind, the more the transpiration rate. So the instrument actually that we use to actually investigate the rate of transpiration is known as a potometer. Okay, and this is what we use to measure the rate of transpiration, right? So if you look at the setup, so this is actually a beaker of water. Then you can see air bubble has been introduced. Okay, this air bubble actually is introduced deliberately because since if we are assuming a transpiration is happening, so the water vapor actually will leave through the leaves. So and we know there has to be this constant pull, transpiration pull hona chahiye, and water should be pulled upward. This is okay. We know the transpiration this is happening. So this movement of air bubble actually will tell us whether at how much rate it is happening. Okay, we look at the distance. Air bubble yahan se leke, if it has moved here, so how much distance it has covered. Then we can calculate it using a formula. Okay, so this is how we actually see the transpiration rate. This part here is actually like we can use it once you're done with an experiment. So you can use this range to bring this air bubble back to the original position. Then you can take another reading. Okay, so this is how this potometer works, right? So this is another view. Generally, the liquid that we take it inside, actually we take it as a dyed liquid, so that अगर air bubble move कर भी रहा है, तो you can actually see, look at the difference कि वो liquid water कितना move किया, because transparent में तो you cannot tell. But अगर वो dyed है, so it's easy for you to identify, right? So this here, what they've shown you is actually is a bubble potometer. Bubble इसलिए कह रहे हैं because that air bubble has been introduced there, and we see कि अगर for example कि this was your initial point where the air bubble was. So then you can measure it through the scale, how much the distance was traveled. So this is how we calculate how much is the rate of transpiration, right? All right, so basic theory क्या है इस potometer use करने के लिए, ठीक है? So the theory is that as the water evaporates from the leaves of the shoot, ठीक है, we know it is definitely moved up the shoot. अब कौन-कौन सी forces act कर रही हैं moving it up? Actually, we know कि there are cohesive forces between the water molecules itself, ठीक है, and adhesive forces are because xylem के साथ से water molecules attached हैं, ठीक है. So what is moving up the chute through the cohesive adhesive forces both, and this will actually make the air bubble to draw along the capillary tube, ठीक है. And definitely this is what we are assuming, it will move at the same rate, ठीक है, जिस rate पे transpiration हो रही है उसी rate पे we'll see that this air bubble is actually moving, ठीक है. However, the thing is that it is not actually solely because of transpiration, ठीक है, that the rate of water uptake being measured is not the same as transpiration. Remember that, because it is not only transpiration that is happening in the plant. Water की requirement बहुत सारी जगहों पर है. Photosynthesis हो रही है, ठीक है, so water requirement वहाँ पर है. Aerobic cellular respiration हो रही है, तो उस केस पे water is actually being produced as a waste product there. So ठीक है, तो a lot of things are actually, it is not 100% transpiration rate नहीं हम actually इसमें calculate कर पाते हैं, ठीक है. But generally like क्योंकि just to understand the fact, हम definitely use करते हैं potometer को, ठीक है. But ये आपको 100% नहीं बता रहा था कि this is not the 100% transpiration जो आप rate calculate करेंगे, ठीक है. So that's see. So if we talk about this mass potometer, it actually measures the water loss from the plant, ठीक है. So water loss from the plant, remember, is not by transpiration only, ठीक है. So हम बहुत सारी चीजें इसके अंदर देख रहे होते हैं. If you talk about the accuracy, क्योंकि again अगर आपको accurate result चाहिए तो जो आप balance use कर रहे हो तो उसकी accuracy के अंदर definitely there's this limitation, ठीक है. And then what else here? What else you are saying? You're saying कि yes, कि since here in the potometer we are assuming that all the water taken up is lost in the transpiration, but remember photosynthesis के अंदर भी water का use हो रहा है. It is used in maintaining turgidity of the cell. Hydrolysis reactions are taking place inside the cell as well, ठीक है. अगर starch है, ठीक है, and it is being used then to be broken down into glucose, eventually aerobic cellular respiration में use हो रहा है. Some of the water is evaporating in the form of respiration, ठीक है. और reasons भी हो सकते हैं कि some water may evaporate because if the apparatus is not sealed. So these are a few formulas that can be used to actually measure all these rates. So for example, if you're measuring the volume of water uptake by the chute, let's say, so this is the formula that you're going to use, ठीक है, because you're using this thin capillary, ठीक है. तो उसका क्रॉस सेक्शनल एरिया अगर हम लेंगे पाई आर स्क्वायर्ड टाइम्स द डिस्टेंस ट्रेवल, तो डिस्टेंस ट्रेवल हम देख रहे हैं ऑफ द एयर बबल, ठीक है, हाउ मच डिस्टेंस हैज इट ट्रेवल्ड. तो एक सेट पॉइंट हमने रखा हुआ होगा, सो यू मल्टीप्लाई द क्रॉस सेक्शनल एरिया बाय द डिस्टेंस, ठीक है, एंड दिस विल एक्चुअली टेल यू how much volume of water has been taken up by the chute, ठीक है. You can also calculate the rate of transpiration. Rate कैसे calculate करते हैं? So this is the distance traveled by the bubble divided by the time taken, ठीक है. So this tells you the rate of transpiration. You can also calculate the rate of water uptake, ठीक है, rate of water uptake, ठीक है, how much like उसकी speed क्या है water uptake की. So there is a speed of movement of air bubble, that is actually this part here, the rate of transpiration times the cross sectional area, ठीक है, and this will actually tell you the rate of water uptake. So a few formulas that can be used to measure, ठीक है. If you want to measure the volume of water uptake, you'll use this formula. If you want to measure the rate of transpiration, you're going to use this formula. And if you want to calculate the rate of water uptake, okay, so this formula here. Right? Now, again, in your paper, you also get a 5 to 6 mark question about how to set up a potometer. So let's go through these. Okay, so again, I'm saying it has to be in a logical order. आपका answer के अंदर एक proper sequence होना चाहिए. So what do we do? Since we are taking a leafy shoot, okay, so we cut it from the plant, but then it has to be actually kept underwater. ये पूरा जब experiment करते, it has to be done under water. जो shoot है, it should be under water. ठीक है, और उसके बाद जब आपने कट किया है तो फिर then you cut the stem as well, उसको भी पानी के अंदर काटना होता है. उसका मेन रीजन बेसिकली ये कि अगर आप उसे बाहर काट रहे हो तो एयर उसके अंदर एंटर हो जाएगा एंड इट विल एक्चुअली जो वाटर कॉलम एक्चुअली जो बना हुआ है वो डिस्ट्रक्ट हो जाएगा. ठीक है, सो मेक श्योर कि हमने जब शूट काटा है प्लांट के अंदर से, ठीक है, यू इमीडिएटली पुट इट अंडर वाटर एंड उसके अंदर से अब एक स्मॉल सेक्शन अब स्टेम का काट रहे हो तो इट शुड बी आल्सो बी डन अंडर वाटर. दिस प्रिवेंट्स द एयर फ्रॉम गेटिंग इन द जाइलम, बिकॉज अगर एयर चली गई तो वाटर कॉलम ब्रेक हो जाएगा. दिस इज व्हाट दैट वी डू नॉट वांट. हम तो कैलकुलेटरी रेट ऑफ ट्रांसपिरेशन कर रहे हैं. ठीक है, बट इन ऑल ऑफ द प्रोसेस मेक श्योर लीव्स वेट नहीं होने चाहिए. ठीक है, बिकॉज अगर लीव्स वेट हो गए सो देन यू कैन अगेन थिंक अबाउट इट कि अब वहां पे वाटर पोटेंशियल का डिफरेंस आ जाएगा तो स्टोमा क्लोज हो जाएगा. ठीक है, और फिर हम रेट ऑफ ट्रांसपिरेशन सही से कैलकुलेट नहीं कर पाएंगे. सो डू नॉट गेट द लीव्स वेट एंड दिस विल अफेक्ट द ट्रांसपिरेशन रेट. ठीक है, देन यू हैव टू पुट द पोटोमीटर अंडर द वाटर. द वाटर नीड्स टू बी फ्रेशली बॉइल्ड एंड कूल्ड. ठीक है, एंड इसका रीजन बेसिकली रिड्यूसेस और मिनिमाइजेस द एयर बबल्स. ठीक है, एंड वाटर के अंदर डालने के बाद भी हैव टू मूव इट फ्रॉम साइड टू साइड. ठीक है, बिकॉज हम कोशिश ये करें कि जितना भी एयर है उस पूरे सिस्टम में वो रिमूव हो जाए. ठीक है, आफ्टर दैट पोटोमीटर को वाटर के अंदर रखने के बाद देन मूविंग इट फ्रॉम साइड टू साइड टू रिमूव as much as possible. Now you're going to attach the leafy shoot into the potometer under water as well. ठीक है, then you try to seal all the joints, ठीक है, with a waterproofing substance like petroleum jelly, so that any water loss is from the leaves only. ठीक है, because this is what we are trying to investigate. Transpiration कैलकुलेट करने की कोशिश करें तो दिस इज व्हाट वी नीड टू सी. सिर्फ लीव्स से होनी चाहिए. तो हम जितने भी ओपनिंग्स हैं जहां जहां से वाटर लॉस हो सकता है उन सब चीजों को आप ब्लॉक करेंगे. देन यू इंश्योर ऑल द एनवायरमेंटल कंडीशंस डू नॉट चेंज around the leaves as this will heavily influence the transpiration rate. ठीक है, so as we talked about the factors, हमने temperature की बात की थी, हमने wind की बात की थी, ठीक है, humidity की बात की थी. तो make sure these environmental factors do not change. Then the air bubble introduced into the capillary tube can be timed as it moves across a set distance. Thus the rate of water loss can be measured. Right, so these are the like steps that you can follow while you're setting up a potometer. Okay, and for this I want you to have a look at this video tutorial about it is shown that how the potometer is set and all the points about that. So have a look at that and then we'll move on to the next topic then.
Okay, I'm going to show you today how to investigate transpiration. I'd like to remind you what transpiration is. It's the evaporation of water from leaves. And also it's important that you remember that transpiration is actually to do with water moving through leaves and out of stomata down a water potential gradient. Whenever water moves in biology, water potential gradients are involved. Now then, a potometer is quite a simple piece of apparatus, but rather tricky to set up and rather tricky to get it working properly. Okay, in its simplest form, it's actually a glass tube. Here is a capillary tube with a scale on it. At one end, we insert a leafy cutting from a plant. Okay, it has to be a nice, healthy leafy cutting. So here I've got a piece of laurel, which will be inserted in there. The whole of the apparatus is full of water. That's represented by the brown here. There's also a syringe or some form of reservoir. This is simply to refill the apparatus after you've used it in an experiment. Now, the idea here is that as the plant loses water by transpiration, a continuous stream of water should be drawn up through the apparatus and then out into the atmosphere through the plant. The rate of transpiration can therefore be measured by measuring the time it takes for the capillary to move a certain distance, or, you know, you could do the opposite and measure the distance moved in a particular time. And that will actually give you a rough guide of the rate of transpiration. Now, unfortunately, the potometer is a little bit tricky to set up, and there are a number of precautions that must be taken if we're going to obtain valid results. Now, remember, valid results are results that have only been affected by the variable being investigated, not other variables, you know, like changes in temperature, changes in light intensity that are going on while you're investigating something else. Now, with a potometer, In order to get valid results, first of all, we've got to make sure that our plant shoot is freshly cut and very healthy. So as soon as you cut that, you stand it in a beaker of water, and before you set up the apparatus, you actually cut that underneath water. Now, that's the second point here, but actually it's quite fiddly, because the whole of the apparatus should really be submerged in a sink or a bucket of water. The leafy shoot should then be cut, freshly cut under the water, and the whole apparatus should be set up underwater. This is actually to stop air bubbles from accessing the apparatus or from being drawn in to the xylem vessels there at the bottom of the stem. Okay, because they will actually break the the column of water and interfere with the rate of transpiration. So it's got to be a healthy shoot, it's got to be cut underwater to stop bubbles from entering, and the whole apparatus has got to be set up underwater to prevent bubbles from entering the apparatus. The leaves have got to be dry before you actually use the potometer, because otherwise water is simply going to block the stomata, mainly on the underside of leaves, and prevent evaporation or transpiration. Now, another thing about this apparatus, you've got to make sure, and you can see all these bits of blue tack and putty and things that have been put on over the years, you have to make sure the whole thing is airtight, okay? And where the shoot is inserted, you need to seal around it very carefully with Vaseline to make sure that when the water is being drawn up through the plant, there's no air being drawn in from outside. Remember, once you've actually finished a run of this, an investigation, you can actually just open a tap or use your syringe to refill, in other words, to push the bubble back to the beginning. So that, in its simple form, is a potometer. Okay, now we can use this to investigate quite a few different independent variables, factors that may affect the rate of transpiration. We could vary the temperature. That's not easy to do in a science lab, but, it can be done. Light intensity is very easy to investigate because we can use a simple bench lamp, okay, to actually vary the light intensity falling on the leaves, okay, and, if that's all you've got, a bench lamp, you can simply use a meter ruler to actually vary your light intensity by moving the lamp at different distances from the leafy shoot there. Another thing you could investigate, though a little more tricky, is humidity. Set the thing up in different humidity conditions. For that, you might have to sort of construct a plastic bag or a bell jar around and, vary humidity conditions in there. Air movement is something that's easy to investigate, because for that you could actually just use a hairdryer, at different distances from the leaf or at different settings. Okay, and hopefully if you followed all these precautions, you should actually be able to get some valid results that are a fairly decent indication of the rate of transpiration.